My name is Chris Ching, and I'm the co-chair of the ENIGMA-Bipolar Disorder Working Group here at the Imaging Genetics Center, part of the Mark and Mary Stevens Neuroimaging and Informatics Institute at the University of Southern California. And today I'm going to be talking about some of the lessons we've been learning from these large-scale international studies of bipolar disorder in the brain through the ENIGMA Bipolar Disorder Working Group. So we'll start with some acknowledgements because these studies are not possible without the thousands of research participants from around the world and the over 200 ENIGMA Bipolar Working Group scientists who I have the pleasure of representing here today. So first with some terminology. So today when I'll talk about neuroimaging, I'll be referring to human brain MRI. I'll abbreviate bipolar disorder with BD from time to time. I'll be talking about case versus control analyses quite a bit. So this is a type of study design in which we compare cases or participants with a diagnosis of bipolar disorder, and we compare them to controls or individuals without a psychiatric diagnosis to better understand the biological signatures specific to bipolar disorder. I'll be talking about study replication and generalizability. These are foundational aspects of the scientific process. Replication being the consistency of findings across similarly designed research studies using different study populations, and generalizability being study findings that reflect the larger population, allowing for accurate predictions about new unseen participants or patients. And today, when I talk about biomarkers, I'll be referring to an objective body measurement that reliably predicts illness progression and treatment response. Now, today I'll be covering some of the major challenges facing bipolar disorder research. I'll talk a little bit about how we as neuroimagers use MRI to study bipolar disorder, and I'll also spend quite a bit of time talking about findings from the ENIGMA Bipolar Disorder Working Group and how we're trying to join forces with researchers from around the world to improve our understanding of the mechanisms driving this illness. So first, let's start with some major challenges. And this is really the story of missing biomarkers. So bipolar disorder is a chronic episodic illness defined by extreme fluctuations in emotion, mood, thought, behavior, and energy. These days, we diagnose the illness based on a detailed clinical interview with a patient to assess the severity, duration of symptoms and signs. And we know that a great many people are affected worldwide by this illness. Unfortunately, unlike other medical disciplines, there's no laboratory tests that exist to help us diagnose and guide treatment for bipolar disorder. So we know that individuals with the illness can have very different symptom profiles. We sometimes refer to this as patient heterogeneity. We know that bipolar symptoms can overlap with other psychiatric illnesses like major depression. A majority of individuals with bipolar disorder have been misdiagnosed at least once in their lifetime, leading to suboptimal treatment. We know that it can take the better part of a decade on average from when a patient starts experiencing their symptoms to when they're properly diagnosed and receiving treatment. And unfortunately, we also know that treatment efficacy can vary quite widely between individuals. It can require quite a bit of trial and error to get right for each patient. So the ways in which we diagnose bipolar disorder and our classic research design have created some challenges for biomarker discovery. So ideally, we'd be able to find a set of biological measures that might conveniently identify or segregate who has the illness from who doesn't. Now, in the real world, we know that individuals with bipolar disorder have very different symptom profiles, responses to treatments, illness progression. And so for all the measures that we've studied, we continue to see varying degrees of overlap and distinction between individuals with the illness and controls. And so researchers have really struggled to link behavior-based diagnostic criteria that we use for bipolar disorder to link that to underlying potential biomarkers using this classic research study design. This has a lot to do with the heterogeneity that we see, differences between individuals with that same bipolar diagnosis. Now I'm going to talk a little bit about how we use neuroimaging to extract brain measures from MRI. So as I mentioned, when we talk about neuroimaging today, we'll be focusing specifically on MRI. An MRI scanner uses a powerful magnet and radio waves to safely create different types of images that you can think of sort of like as filters on your camera that capture different aspects of brain structure and function. So unlike other techniques, there's no radiation associated with MRI. It's completely safe. We just have to be careful about anyone with any sort of metal implant or medical device, many of which these days are MR safe. But because it's a big magnet, that's the major safety concern. Now, neuroimaging researchers over the past several decades have been developing a wide range of tools to extract brain measures from these different MRI images. So on the left, we see some methods to extract gray matter volume, thickness, surface area measures across different brain regions. Second to left, we have a set of techniques to extract white matter microstructure and connectivity measures between brain regions. In the upper right, we have functional neuroimaging, or a way of quantifying neural activity based on changes in blood oxygen. And in the bottom right, we have a classic MR technique in which we can quantify metabolite concentrations across different brain regions. Now, there are lots of other techniques, many other MR modalities. Today, we'll be focusing mainly on the two on the left, so the gray matter and white matter structural measures. So how can neuroimaging help us fill in the gaps for our knowledge of bipolar disorder? So mental illnesses are brain illnesses, and if we want to study how bipolar disorder works, we need to look at the human brain. And so neuroimaging offers a safe, non-invasive way of measuring subtle changes in the human brain. Now, if we look at other conditions, for example, Alzheimer's disease, it can be visually quite apparent that there are some changes happening in the brain. So this individual has late stage Alzheimer's. You can see quite a bit of significant brain atrophy, especially compared to the healthy individual on the left. And in psychiatric illness, we see that the changes happening throughout the brain are not visually apparent. And in fact, we require using some of these neuroimaging tools to extract those measurements so that we can model these alterations and pull out these subtle effects from these brain images. And that's exactly what neuroimaging researchers have been doing over the past several decades, really providing important insights into that subtle brain alterations that lead to disruptions in emotion and reward processing. So they're the hallmarks of the illness. One of the other things that we're really trying to do is to link what we see on an MRI level to the underlying cellular, molecular, genetic processes to try to reveal illness-specific mechanisms and hopefully new treatment targets. A little bit later, I'll be talking about some of the next generation analysis methods like artificial intelligence, machine learning, and big data approaches that are changing the way we look at biomarkers, not only within bipolar disorder, but across psychiatric illnesses. So there are a number of factors that can threaten the consistency of neuroimaging findings in bipolar disorder. It's been widely reported that there is a crisis of replication and generalizability across much of biomedical research. This isn't just an issue for neuroimaging or psychiatric illness. It really spans much of science. But we know that neuroimaging studies can be challenged in certain ways. For one, neuroimaging is expensive. It can be quite expensive to collect data from participants. And historically, most studies have collected fewer than 100 participants. They often collect just one time point, or a snapshot in time, so they don't follow individuals for months or years. Most studies have been conducted in largely European ancestry samples. And often studies include strict inclusion and exclusion criteria. And that's important for study design, but it can also make it challenging to replicate study findings in independent cohorts. As I mentioned before, researchers can use different data processing and analysis techniques that can also make it challenging to compare findings across studies. And this is all compounded by the fact that we know bipolar disorder involves subtle brain alterations. And when you add that to the variation that we see between individuals, this again can create challenges for study replication and generalizability. And lastly, we know that MRI studies can sometimes have limited interpretability. So few studies have really tried to bridge that gap between what we see on an MRI level to underlying alterations in the cellular, molecular, and genetic mechanisms that might be driving those MR level variations that we see. So the question is there a way forward? So over the years, there have been many independent neuroimaging studies conducted in bipolar disorder. Many of these researchers work obviously independently and don't often pool their data samples. And so the question is, could we potentially boost more diverse samples by combining or reviving this existing data that already exists out in the world? And so in theory, that would improve generalizability, create for more representative samples of the wider population. If we pooled that data, we might be able to boost our modeling or our statistical power by implementing standardized processing and analysis methods, right? So that could also, in theory, improve replication, showing similar results from one study to the next. And then if we were able to integrate these large scale sort of standardized brain metrics with other biological measures, we could potentially use big data techniques to allow us to move beyond our classic case versus control study design and look for more clinically relevant potential biomarkers. So now I'm going to spend the rest of the presentation talking about how the ENIGMA Bipolar Disorder Working Group is trying to do just that, revive and pool existing data samples to boost the replication and generalizability and hopefully drive new insights. Now, ENIGMA stands for Enhancing Neuroimaging Genetics Through Meta-Analysis. This was an effort that started back in 2009 by Paul Thompson, Jason Stein, and others, really aimed at looking at what the genetic factors were that influence brain structure and function. And so to conduct these types of studies, you need lots and lots of data. The effects of common genetic variants on brain structure are quite subtle, and so you need large samples to pull out those subtle associations. And so that's exactly what they did. They went around the world and they were able to convince a whole bunch of researchers to contribute data to this first study, conducted in a sample that was much larger than any one group could acquire on their own. And that first project really sparked a movement that's become the ENIGMA Consortium, a global network of over 2,000 scientists pooling independently collected data samples to study a range of brain conditions. And so one of the questions early on was, well, can we actually combine all these independently collected samples that come from different scanners and use different inclusion and exclusion criteria? And so I like to use an analogy when thinking about how we've been doing this. So let's say we asked a question, who makes the best omelet? And if we look around the world, we sample omelets from across the globe, we see a lot of variation in style, how people actually interpret what an omelet is. And so that might be a little difficult in terms of deciding who a winner might be. And so maybe by providing some basic rules like a standard recipe, we might be able to level the playing field to make judging a winner a bit easier. So if we give people a recipe, we'd expect to get in return are omelets that look a bit more like one another. So what we've done here is we've constrained the variation in style so that we can limit that variation and maybe make judging a winner a bit easier. Now, in the ENIGMA Consortium, we do something very similar. We've basically designed standardized methods or cookbook recipes for each group to go around and re-extract different types of brain measures in a standardized way. Right? So we've standardized the recipe, and we use widely implemented protocols, freely available tools, our own in-house built tools to extract all types of different brain measures, trying to limit the variability across these different samples. Now, the ENIGMA working group consists of over 50 different subgroups. They can broadly be divided into the groups that are developing the algorithms or those standardized cookbook recipes, and then the groups applying those standardized processing and analysis methods to a range of brain conditions that you see here. Now, the ENIGMA Bipolar Disorder Working Group was established in 2012. It now consists of 50 international cohorts, over 200 scientists from 20 countries, 55 research institutions, and a pooled sample of about 3,500 individuals with bipolar disorder and close to 9,000 undiagnosed controls. And I co-chair the working group with my colleague, Dr. Ole Andreasen from the University of Oslo. Now, if people are interested in seeing some more detail about what I'll be talking about, I encourage you to take a look at a recent review article that we published outlining some of the highlights over the past decade and where we're headed in the future. So before we jump into some foundational studies, I kind of want to summarize what I'll be talking about for the rest of the presentation. This is an important slide because it gives you sort of an overall outlook of what we've been finding throughout these ENIGMA working group studies. So since 2012, we published 15 studies, each of them the largest of their kind. We have about 18 ongoing projects. You can see a full list on our website with a description of all these projects. But generally what we've been seeing is that bipolar disorder involves subtle brain alterations. So smaller, thinner gray matter regions, altered white matter integrity, but all subtle alterations. We've shown that there are differential effects of commonly prescribed medications. We've shown some modest predictive power using AI and machine learning techniques. And we're beginning to conduct the largest cross-disorder studies ever. So again, all the results that I'll be showing you in the following slides represent subtle alterations that we're able to pick up by combining these large-scale samples. So in our first foundational study, we were looking at subcortical volumes. So these are deep brain structures. Again, the largest study that had ever been combined across several thousand individuals. And generally what we found were that individuals with bipolar disorder had smaller subcortical volumes in a couple key regions. One of the purposes of this study was to try to drive consensus. Prior studies had shown some variability in some findings. And so we were trying to establish what regions appear to be robustly associated with the illness and also get a better estimate for the subtlety of that effect. Now, in our second study, we looked at cortical structure. So in the bottom right, you can see the highlighted outer sort of ribbon of layered cell bodies called the cortex. And we can measure different aspects of local thickness and surface area from these structures. And so, again, in a boosted sample of over 6,000 individuals, what we found was a widespread and subtle pattern of lower thickness associated with bipolar disorder. Now, one of the things we also looked at was mapping the effects of commonly prescribed medications. And so we found individuals taking lithium tended to have thicker cortex, and those taking anticonvulsants actually had thinner cortex. So I'll talk a little bit more about modeling drug interactions and effects on brain structure a bit later. So in our third sort of foundational study, we were looking at the brain's white matter, so connection between brain regions. And again, we found this widespread, subtle pattern of altered white matter microstructural integrity. We found that lithium appeared to have a normalizing effect on these white matter metrics. And importantly, the patterns that we were seeing here to some extent overlap what we were seeing from some other large-scale ENIGMA studies of schizophrenia and major depression. Now, in our fourth sort of foundational study, what we asked was whether or not we could use these standardized brain metrics and load them into a machine learning or AI algorithm to perform diagnostic predictions. So whether or not we can use these brain measures to tell whether someone was diagnosed with bipolar disorder or if they were a control participant. And what we found was that we topped out at about 65% accuracy in diagnosing bipolar individuals from those not, those without a diagnosis. And so 50% would be a coin flip, chance, guess. So 65% doesn't seem all that impressive, but I think if we look and see that we are using a relatively pragmatic, small set of brain features, and those brain features driving the prediction were both biologically relevant and consistent across study samples. This study really provides an important jumping off point for many of the studies to come and the research for the future in which we're trying to boost this potential performance by adding more brain features, more complex AI models, et cetera. Now, as I started to talk a bit more about some of our ongoing work and future studies, we have a couple guiding principles. So every time we look at a bipolar-related finding, we're constantly asking what are the other brain measures or analysis techniques that we can use to explore or understand what these results mean. We're also trying to look at how brain change over time is associated with illness progression. So moving away from studying just the snapshot in time and really looking at how the brain is changing. We know that bipolar disorder is an episodic illness, and to understand how those episodes work, we really need to look at how an individual's brain might be changing over time. And so we're also trying to move beyond our case versus control classic research design, trying to link individual symptom profiles and other clinical risk factors to brain structure and function, not only within bipolar illness, but across related psychiatric illnesses. And then lastly, a really important effort in trying to bridge that gap. So linking underlying cellular and molecular factors that might help us explain what we see on a higher level brain MRI. Again, not only within bipolar illness, but spanning related illnesses. Now, this is an example of a study in which we were trying to dive a bit deeper into an initial finding. So you may remember in that first foundational study, we found that individuals with bipolar disorder had smaller hippocampal volumes. And so we know this structure is vitally important to learning and memory and involved in the circuitry of emotion and reward processing. We also know that it has functionally distinct subcompartments that we can segment and measure. And so one of the questions we were asking here was to see whether or not bipolar illness might be targeting specific subfields of this structure. Now, what we found was, on average, those with bipolar disorder had lower or smaller subcortical, subregional volumes across the entire hippocampus. But again, we found that lithium appeared to have a normalizing effect on these subregional volumes. Now we have another method, high resolution shape analysis method, where we can model subregional alterations as well. So here we can look at local thickness and surface area variations, not just within the hippocampus, which is structure 4 here, but now we can look at the sort of distribution of effects across other subcortical structures to better understand the underlying circuitry vulnerabilities associated with this illness. So there's evidence to suggest that bipolar disorder may involve advanced or accelerated aging processes. And so Dr. Lisa Eiler and her team at UCSD have been using the ENIGMA-standardized brain measures to create what are known as brain predicted age difference models. So what you can do is you can take your control subjects, those not diagnosed with bipolar disorder, and you can load them into an algorithm that will be tuned to predicting a person's age based on what their brain looks like. And these algorithms can get quite accurate at predicting a person's chronological age. Now, the next thing you can do after you build your model is then load in the individuals with a bipolar diagnosis. So again, making use of the large-scale ENIGMA data. And the model will take their brain and predict how old they are. Now, if the model predicts that an individual is older than their actual chronological age, we call this having older brain age. Now, if the model predicts an individual as being younger than their actual chronological age, we call this younger brain age. And so Dr. Eiler and her team have been looking at associating these brain pad scores with different clinical characteristics, including course of illness, symptom severity, medication status. And on this plot, the bottom left, we see in pink that the individuals with bipolar disorder have slightly higher brain pad scores. Their brains appear slightly older compared to the undiagnosed individuals. And so this measure may be another way, an interesting way, of potentially parsing some of that patient heterogeneity that I talked about before. Now, one of the things we did last year is we published the largest longitudinal study of brain change ever conducted in bipolar disorder. One thing you'll notice here is the sample sizes are quite modest. So I showed you some studies before where we were comparing thousands of participants with bipolar disorder versus controls. Here we have a much more modest sample, and that's because there have been very few studies looking at following individuals over time to look at how their brain changes, partly because of expense, partly because some of those limiting factors that I talked about before. But in this large scale study in which we computed yearly rates of brain change, we found that bipolar illness was associated with, again, very subtle longitudinal brain changes over time. And in fact, some regions actually showed faster decline in the control participants. Now, we also showed that a greater number of hypo and manic episodes was associated with faster cortical thinning in certain regions. And this potentially suggests the importance of treating and maintaining those fluctuating mood symptoms to potentially improve long-term brain health. Now, one of the other things that we've been trying to do is look at how potentially modifiable clinical factors like obesity might influence the way we understand bipolar-related brain signatures. So in this study, combining data from 17 ENIGMA bipolar sites, we found that on average, individuals with bipolar disorder tended to have higher BMI, body mass index, compared to controls. And this is for a number of well-documented clinical reasons, including side effects of commonly prescribed medications. But interestingly, what we found was that higher BMI was associated with similar regional brain alterations as what we saw in bipolar disorder, and could be partially accounting for some of the effects that we saw in bipolar illness. So this points to an important idea in that we need to model some of these other clinical factors to better understand the extent to which the illness is affecting brain structure and function, and how those other clinical factors may accentuate some of those alterations. Now, the ENIGMA Bipolar Working Group is also trying to rethink the way we model medication-brain interactions. So as I had shown before, we found that commonly prescribed medications can differentially affect brain structure and function, and that can complicate our understanding of the illness-specific brain alterations. We also know that most patients with bipolar disorder are taking more than one medication. And so we really need more complex polypharmacy models to try to link the complex brain alterations to treatments, and especially to treatment response. And so how we've been doing this over the past several years is taking a classic modeling approach in which we try to associate particular drug classes with alterations in brain structure. And so what we tend to find is, as I showed before, lithium tends to make brain structures larger, thicker. We see less disrupted white matter. But when it comes to anti-epileptic or antipsychotic medications, we tend to see the opposite effect. So smaller subcortical volumes, thinner cortex, more disrupted white matter. But we know that there's a better way of doing this modeling. And so what we've been doing now and moving into the future is really disaggregating some of these drug categories and pulling out the actual drugs that individuals are taking and looking at combining drugs based on their known pharmacological mode of action, right? So really trying to tie how a drug works on a cellular level to these brain alterations. And so combining other detailed information like the dose, duration of treatment, other substance use, we're hoping that we can get a better understanding of how these different medications affect brain structure and function, and potentially, hopefully, try to be able to predict treatment response, right? So with the ultimate goal of trying to drive down or limit that trial and error that we see when treating patients today. Now, one of the things that's really beneficial about the ENIGMA approach is that because all the groups are using the same sort of cookbook recipes and performing similar types of analyses, we can begin to compare results across different working group studies. And so here we see some results from the ENIGMA Major Depression Working Group. These maps and figures look probably very similar to what I was showing you before from our bipolar working group. And that's because they're conducting very similar analyses using the same standardized brain measures. And so one of the things we can do is to begin to compare these large-scale standardized brain metrics really on an unprecedented scale. So each of these brains here represent the largest case versus control published studies looking at cortical thickness in these different brain conditions. And so one of the things we're doing now is to move towards actually directly comparing these measures across these different working group studies. So one of the projects we have underway is combining data from the ENIGMA Bipolar, Schizophrenia, and Major Depression working groups. We know these conditions have underlying or overlapping symptoms, overlapping responses to treatments, and similar genetic risk factors. And so by directly comparing these standardized brain measures across these disorders, really trying to map the overlapping and distinct brain patterns that we see, as well as combining the data. So if we pool these data samples together and we ignore the diagnosis, we might be able to pick up brain features that predict more clinically interesting factors like symptom profiles, treatments, duration and severity of illness, et cetera. So in an effort to look across disorders and try to bridge that gap to the underlying biology, in a recent study, we looked at the similarity in brain signatures across these ENIGMA psychiatric studies, to some extent mirrored the underlying genetic similarities we see of the same disorders based on genome-wide association studies. Now, this is important because we know that many of these illnesses have strong genetic components. And so here we're showing that the underlying genetic architecture appears to reflect what we see on a higher level MRI scale. Now, another way of looking at bridging this potential gap, work from Thomas Paus and Yash Patel, in which they've published 2 studies now trying to link brain MRI signatures from the ENIGMA bipolar and other psychiatric working groups to underlying patterns of gene expression. So what they have been doing is putting together these large cross-disorder brain signatures from the ENIGMA working group. And by linking those to human brain gene expression database information, we're finding is that there are some shared and distinct cell-specific gene expression patterns that may be related to cross-disorder brain signatures, right? So pointing to potential future hypotheses that we can test regarding the mechanisms that might be driving brain change and risk for developing these different mental illnesses. Now, we also have something called the ENIGMA Toolbox. So again, a tool trying to bridge that gap between the macro and micro. This is an online toolkit for exploring MRI results from the ENIGMA working groups, along with some really interesting cellular and molecular databases. This is a tool that we use in research, but it's also freely available online. And so if there are data scientists, hobbyists out there, you are free to be making discoveries right alongside us. So where are we headed in terms of the ENIGMA bipolar working group? So we're really, in the next decade to come, looking to grow our samples of standardized brain modalities. That includes both structural and functional brain connectivity, brain metabolites. We're now looking back at understudied brain regions with these large-scale samples. We're also looking at adding these higher resolution features, looking at aspects of asymmetry, lots of really interesting projects. And you can find more information about that on our website. We're also trying to extend our analyses to more detailed clinical and genetic factors. That includes information on symptoms, medications, substance use, and other risk factors. We're also looking at really trying to combine genetic information with these standardized brain data sets. So looking at how polygenic risk affects brain structure and function. And we have another set of really exciting studies looking at unaffected first-degree relatives of individuals with bipolar disorder, really to try to pull apart what we think are the strong sort of genetic factors that influence this illness. As I mentioned before, we're also enhancing our analysis methods, right? We're trying to look at longitudinal brain change modeling, applying some of these deep learning and other big data approaches by combining these large samples that span classic disorders with other sort of biological data alongside the standardized brain measures we've been generating. We're hoping to provide or model more clinically useful data-driven potential categories that might indicate subgroups of individuals across disorders that are less or more likely to respond to particular medications. They may fall into higher risk for cognitive decline or suicide, et cetera. Now, this is an example of one of the deep learning tools that we've been using and some of the more high resolution brain shape features that we're feeding into these pipelines to try to boost our performance of some of these predictive models. And so as I showed before in one of those foundational studies, our first study to use machine learning, in which we topped out at about 65% accuracy to tell the difference between individuals with bipolar disorder versus controls. These methods may help us boost that performance, and not just performing diagnostic classification, but also trying to predict duration and severity of illness, symptom profiles, medication responses, and more. Now, one of the important collaborations I wanted to mention here is some work we're doing with the Psychiatric Genomics Consortium bipolar working group. And so the PGC is really a similar type of consortium, an international study combining samples all around the world to find novel genetic risk markers for bipolar disorder. And because many of the ENIGMA samples also have genetic data, we're looking at how an individual's genetic risk for bipolar disorder influences their brain structure and function, and whether or not we can use the genetic brain links to help inform future clinical decisions. Now, in summary, I'd like to hope that I conveyed that it's never too late to learn new things from old data. I think we've been showing over this past decade that the ENIGMA approach to team science can really work, that these large-scale analyses can help drive consensus as well as novel findings while improving our replication and generalizability of these studies. Again, emphasizing that the brain alterations that we see in bipolar disorder are subtle. They appear to be influenced by many different complicated clinical factors, including treatment, that we continue to try to model in more complex ways. We're also thinking that these cross-disorder studies that I shared with you are a real promising path forward for potentially discovering new biomarkers for psychiatric illness. And lastly, I hope that I emphasize our efforts in trying to bridge that gap between what we see on an MRI level to the underlying microbiological cells, so that we can better understand the mechanisms driving what we see on the brain MRI. Now, in closing, I'd like to bring us back to the ENIGMA Bipolar Disorder Working Group map. Again, these are all the sites that have come together from around the world to build the most diverse, large-scale analyses of the brain in bipolar disorder. But I think it's important that we're honest about the glaring omissions that we see from this map. And while we'll probably never be able to collect MRI samples from all the points along this map, we'll continue to strive to build the most diverse samples to improve our replication and generalizability of our study findings, so that they truly apply to bipolar disorder as it appears across the world, across countries, across ancestry, more. So we like to end with an invitation. If you know of groups that aren't participating in this effort yet, please do reach out. We look forward to hearing from you. And with that, I'd like to close with some acknowledgements. Again, back to the study participants. These studies would not be possible without all of their efforts and all the efforts from the ENIGMA Bipolar Working Group scientists from around the world. And then lastly, some funding acknowledgements, some recent funding from the National Institute of Mental Health, as well as the Bazuki Brain Research Fund and the Milken Institute Center for Strategic Philanthropy, helping us with the medications initiative that we have ongoing. Thank you very much.